Today we have a really great treat. Professor Josien van Wolfswinkel, if I said that right, I think it's just a great name, who did her undergraduate at Utrecht and her graduate work there also. Got her PhD and then worked at the Whitehead Institute up at MIT, and then we've been fortunate to have her as a new faculty member for the last 5-6 years or so, something, 4 years, right? And she does some really interesting work in RNA biology, and she's going to tell us a little bit about some of the things that are related and how she became a scientist. So join me in welcoming Professor Wolfswinkel.
So thank you for the nice introduction. I want to start out by thanking or asking for a big round of applause for all the volunteers who've helped set up all these demos and for Professor Josien for organizing all of this. I hope you've enjoyed them all, so I want to take a moment to thank them all for that. So my name is Josien, and I am a professor in biology in one of the biology departments here. And that means that I teach the students here, and that I also have a group of people that we run a lab with. So we have a group of people that together we do research with. And today I'm going to tell you a little bit about what that research is that we're doing and why we're doing this. Well, as I said, I'm a biologist, and so that means the study of living things. And the way we study biology is in a large part by observing. So we look at biology as it happens in real life. We look at it, observe it in the wild. We also bring it into the lab and use microscopes to look at the organisms that we study. And then we do experiments and do analysis and try to understand how things actually work. And to tell you a little bit about how I got into this, as Professor Josien mentioned, I am originally not from the US. I'm originally from Netherlands. This is a tiny country in Europe, over there. I think it's smaller than Connecticut. It's definitely not that much larger. You may have heard of it because of its tulips. It exports a lot of tulips. It has these beautiful old cities with canals and really steep houses and lots of bikes. And so I grew up there in, let's say, the 80s. This is me when I was about 7 years old with my brother and sister and cousins. And we lived in a smaller city in the Netherlands, and there was a lot of parks around. And I'd say we were always interested in anything that lived around us. We would always drag things in that we felt needed help. So here we found a nest of eggs and a bird that a duck that was hatching these eggs. And so every day we'd feed the bird and then check whether the eggs were still there and how it was going, how it was progressing. Also any other animals. So if there were stray cats around, we would always manage to drag them home and help grow them up. Many times we had many different types of animals that didn't necessarily go well together, like birds that fell out of the nest together with the cats, together with a rabbit that fell in a ditch, and tried to all nurse them back. So in some way I was always interested in the things that were living around me. But that doesn't necessarily mean that I always knew I wanted to be a biologist. In fact, when I was in school, I enjoyed a lot of the science subjects. I really liked math. I really enjoyed physics, and I really enjoyed chemistry, and I thought all these things were very beautiful. There were systems where you can learn to understand, learn to do calculations, and if they are very neat in a way that if you know how to do a calculation, you can solve an equation. If you know how forces work, you can understand how a spring can move back and forth. And if you understand a bit about chemistry, you can understand how molecules can come together and what kind of reactions they would do. And to me, actually, at that time, biology was a little bit fuzzy. It seemed that it was very difficult to understand. And it took me all the way until the end of high school, actually, to realize that the reason biology is so fuzzy is because it's very complicated. And that it takes all these things that we learn in math, all these principles from math and from physics and chemistry, that they're all true, but they're all combined in biology. They all work together. And together they create these things that are very meaningful to us, the things that we like to eat, the creatures that we like to call our friends, and actually make up the complex ecosystems around us that are both very important to us, but also shape the world as we know it. And so that actually that thing about biology that in some way was the most complicated or the most difficult to me is actually what in the end had the most value to me, of how complex these things are that are together and how delicate it actually is that this works. And so to emphasize that a little bit more and to think about how diverse biology actually is, I want to go for a couple of examples of how wide and vast that diversity is and how beautiful some of these things are. So one of the things to think about in biology is scale. So just the size of things. This is a scale that is drawn in meters. So we are somewhere between 1 and 2 meters typically. But there's animals that are much, much larger, that like whales can be tens and tens of meters long. On the other hand, we know of many organisms that are smaller. We know if we go to the beach and we look at shells that we find that are typically about a centimeter or so. So a centimeter is if you look at your index finger and you take that little bit from the last joint to the end of your finger, that's about a centimeter. But there's many things that are much, much smaller. So if you would divide that into 10 little pieces, then you get to a millimeter. Lots of animals are around that size. If you even divide that 1,000 times more into tiny little pieces, you get to the size of microbes. So really, really small things can be alive. And on the other hand, really, really big things are there in biology as well. Take another example. How fast can things go? There's a huge variety. So cheetahs that can go 75 miles per hour, which is as fast as your car drives on the highway. And they have an enormous amount of muscle that they use to be able to achieve that. On the other hand, sloths, although they look very cute, they're very, very slow, and they have used their energy in a very different way. So they optimize to, well, be very efficient with their energy, but then are very slow in their movements. And there's animals like, for example, hummingbirds that can flap their wings 70 times per second. Just to imagine, this is 1, 2, 70 times in every beat. Just give it well, you can give it a try. We won't be able to do it. Other animals, for example, a recent story was about this blind cave salamander that actually sat still for 7 years. So it doesn't move, barely moves at all. Huge variety again. Also in how long things can live. So, for example, mayflies, we know, live only for a day. But tortoises can live for well over 100 years. And even apart from that, when we look at the way biology looks, it can also create some of the prettiest colors and prettiest shapes that we can imagine. Some of those examples are here. Some of the prettiest birds with feathers that make all these beautiful colors. Structures that animals can make. For example, some that have sticking features that they can use to get prey in, or very thin tentacles with stinging cells that they can use as little harpoons to shoot and catch things. Beautiful structures with which they can filter water, beautiful fins. All these very complicated structures and beautiful colors are there throughout biology. So if biology is so diverse, and if it can look in take so many different shapes, how can we even think that we might understand any of this? Because if everything is so completely different, where would we even start? And so the answer to that is actually just as like you would do with a giant jigsaw puzzle. We just do it one piece at a time. You find one little bit that you can figure out, and that's where you start. And then from there, we try to build. And that's actually exactly what we did in biology. And the way we've started to try and understand about how biology works and about how all this diversity comes about is by picking a set of organisms, a set of systems that we study. And those we call model organisms. And we picked things that actually are all around us and that we very commonly come across. We picked yeast, which is the organism that we use to make bread and make beer. It has been with us for thousands of years. We know how to grow it. It's been with us forever. Fruit flies, if you ever have had some very ripe bananas or apples or basically any type of fruit, you'll know that these magically come. You don't really have to do anything to attract them, you'll find them. Arabidopsis, this is a weed that might grow in your yard and grows all around. Mice, if you've ever lived in an old house, you know that they'll find you. And these are little worms that live in the soil everywhere. And later on, we also found a fish that we use as a model system. And so these are examples of biology. Don't represent all of it, but we picked a couple of places to start, a couple of puzzle pieces to start with. And from that, what we found is something that we call biological conservation. And this is a very important concept So I'll take a moment to explain that. What we found between these very different systems is that actually they work in a very similar way. And in a way that is so similar that it cannot be a coincidence. Let me explain it in a different way. How many of you know the song Twinkle, Twinkle, Little Star? All of you. Great. So imagine you might have, as a child, have heard your mother sing this to you. You might have heard a teacher in school sing this to you. And you might have gone to the playground and hear a father sing it to their child. And so one possibility would be that all those people, your mother, your teacher, the father in the playground, that they all came up with the same words and the same melody by themselves and started singing this song. So who of you think that this is a real possibility? So very few. So I would say that probably nobody thinks that that is what happened. Does anybody have a theory? How could this have happened? Do you want to? Yeah?
Somebody taught them?
Somebody taught them, exactly. There has been somebody in the past who came up with this song, who came up with these lyrics, and we may not know anymore who that was, what this person looked like, when this was. This person may not be here around us anymore, but they came up with this song and they used it and they taught it to some people and they taught it to other people, and this is how it spread. And this is exactly how we think about the similarities in biology. Some of these things that these organisms are actually all the principles that these organisms use are so similar, and they use them in exactly the same way, that this is not by coincidence. This is because there was an organism long, long time ago that's not with us anymore, but was there hundreds of millions of years ago that used these principles for the first time, and they're spread through all the organisms around us. And so this is a very important concept of what we found. But let me explain to you a little bit more about how deep that conservation goes. So in the first place, all organisms that we know of, all living organisms, are made out of cells. And cells are basically bubbles. They're membranes around a piece of space where the reaction conditions, where conditions are very stable, and which make up all these living organisms. And within these cells, there's all kinds of other structures, often surrounded by membranes, that help these units to make energy, to make components, and that are important for the functioning. And one of these very important components is what we call the nucleus, or the core of the cell. This nucleus is so important because it contains some of the material that is, we call it DNA. So every cell contains a nucleus, and in the nucleus are what we call chromosomes. And the chromosomes are actually really, really, really long strands of DNA that are packed together in there. If you put all the DNA of one of our cells and stretch it out, it would actually be 6 feet long. So this is really a long amount of DNA, and that is all packed into this one little nucleus. So when we look at the DNA, what it actually is, it's a really long string, as I said, and it contains, the string itself is made out of a backbone, a sugar backbone, and there's 4 different types of beads, which are the bases. So we have a C, a G, an A, and a T. And so it's a really long string with these 4 different colors that can be in any kind of order, and there is millions and millions of these bases together that make up the sequence of our DNA. So the DNA actually is not just this one single strand, it has a very good trick to it, and that is that it's double-stranded. So there's 2 strands that circle around each other, we call a double helix, and there's always 2 bases that go together. So an A always wants to go with a T, and a C always wants to go with a G. So these 2 are actually the same kind of information is in there twice, because if we know that there's a T here, we know there has to be an A in the other one. And this makes this very stable, and makes that the information is actually stored in a very reliable way. That even if something would go wrong, if, for example, a base would flip out, we actually know how to repair it, because we know exactly how to get that base back and what base that should be. So this information, the fact that this is double-stranded, is actually actively used by the cell, because cells, one of the core things about life is that things have to multiply, have to be able to persist for a long time. And what the cells will do is that they will duplicate their DNA. And the way that works is that you can separate the two strands. And now you know for each of the two strands what it has to make. You know that this one, where the T was, it will have to make an A. Where the G was, it will have to make a C. And so we can just go on and make that second strand. And the other strand that we've separated, we can do the exact same thing. We can just make them. And now we have two copies that are completely identical. And this is the way that information is actually multiplied in an organism. This is used a lot in biology. Every time a cell duplicates, it duplicates its DNA. And so this is a process that we call the cell cycle. So if we start out with a cell that has two strands of DNA, two chromosomes in here, the first thing it will do when it wants to amplify or multiply is that it does a process called DNA replication. And so it duplicates its chromosomes. So this is exactly the process we just looked at of duplicating the information. And then these cells go through a process where they align these chromosomes in a way that they can separate them so that the information will be split evenly between the two different partitions. And then the cell can divide. And these two cells that it's dividing into will now have the exact same information. So all the information is copied. And this is at the core of the development of every organism, actually, of every multicellular organism. Because as you may know, we as organisms and also other animals and other plants, we all originally come from one cell. We all start out as one egg cell and one sperm cell coming together. And that activates some important processes. But this one individual cell then starts to divide. It makes more and more and more cells that over time start to make structures, make different shapes, start to differentiate, as we call it, to make different types of cells, and eventually make an entire organism. So this means that all our cells, all the cells in our body, are derived from this one single cell, and they all contain the exact same DNA molecules, the exact same sequences. So what is in this DNA that is so important? Why do we want to keep it around all the time? Well, what it is actually lots and lots and lots of these bases, lots and lots and lots of letters. Our genome contains 300 billion of them. And so this is an enormous amount of information. A whole bunch of it, we don't exactly know what it is. But there are some parts of it where there is a kind of alert sequence. It's a kind of little sign like, now pay attention. And this is what we call a promoter. And this is the start of a gene. A gene is then a piece of the DNA where a copy can be made, and that actually helps to make a protein. So the DNA by itself is just the information. It doesn't really do anything. But the gene is the recipe to make a protein, and that makes all the functions in the cell. So what this alert sequence, this promoter does, is alert the cell and get a complex we call an RNA polymerase to make a little copy that we call RNA. And that copy then is the recipe to make a protein. So the genes are actually the instructions. These are the units that are very important to a cell. And we can see them as kind of the recipes. So they are telling the cell what to make and how to make it. And these proteins then are the structures, make up all the structures in the cell and do all the things in these cells. So not only this is what we found as one of the things that is widely conserved. All organisms have cells, all organisms have DNA, all organisms make RNA and make proteins and do this in the same way. But it goes deeper than that. Actually, when we look at these genes, at these instructions, it turns out those are also very widely conserved. So they're also very similar between different organisms. One example for this, for example, is actin. This is a structure that is in the cells. It's these green fibers here. And that makes what we call the cytoskeleton, so the skeleton of the cell. It makes the structures, it helps the tension of the cells. These fibers are actually not one thing. Every single fiber is made out of lots of tiny little units. So in that way, it's a little bit like the DNA. It's lots of little beads. In the actin, it's a lot of little proteins that are making them together. And this is what that looks like. So these are these individual proteins. Every single yellow one here is an individual protein. And these can come together and can build a chain, and it always grows in the same direction, and so it gets longer and longer. And actually, while it's growing, it changes its state a little bit, and so it can also, well, depolymerize, so fall apart again on the other side. And this is an extremely important thing for the cell because it allows the cell to build structures, to put out fibers, to move. This is an example of a cell like that, that because of the actin is able to put out feelers and to feel around and to actually move its entire body over. And this is what happens in our bodies too. There are cells that are on the move, cells, for example, in our bloodstream that have to get out of the bloodstream, that are moving and using these fibers to move around. So now if we look at this actin, at this protein, and look at it in different systems, we can look at the actin from a mouse or the actin from a microbe, from one of these really, really tiny micrometer small organisms. And when we look at what their proteins look like, they actually look exactly the same. If we overlay them, we can barely see the difference. They make the exact same structure. And not only do they look the same, they function the same. So they also make these polymers and they help protrude these fibers. So this is a very important protein for all cells. It goes even further than that. So I told you about RNA polymerase that is necessary to make RNA and to make protein. And so RNA polymerase itself actually is a protein, and so it's very well conserved. It is a gene. But one thing you may realize is that I've skipped over something. I've told you that all the cells in our body have the same DNA, have the exact same genes. But we know that not all our cells look exactly the same way, right? We know that there's cells, for example, in that our brain cells, our neurons, are able to signal. They're able to guide electrical pulses, and they have really long connections so that we can feel something that's happening in our toe and signal that through to the brain. We also know that, for example, the muscles in our stomach can contract. These have very strong fibers, and they are very good at contracting, which a neuron would never do. Or the epithelium in our intestine. So these are cells that are made for taking up material. And they have, again, a very different morphology. They look very, very different, and they function very different. So if they have the exact same DNA and the exact same genes, how can it be that they come out in such a very different way? Well, that is because I skipped over a little bit of this expression of these genes, about the reading of these genes. So I told you that there's these promoters that then are a signal for instructions to come. And the fact is, if that promoter is there all by itself and RNA polymerase is allowed to bind there and do something, actually it does very, very little by itself. It's not very active. There are even some proteins that can bind in the region that we call inhibitors, and they switch it off completely. After that, it would not be active at all anymore. But there are other proteins, if we actually really want to have a gene, have the instructions being read, we have proteins called transcription factors that can bind the DNA and signal to the polymerase that this is a gene that has to be used. And they will help to activate the polymerase, to make the RNA, to make the protein, and then the gene is actually on. And so different cell types will have different transcription factors. These transcription factors, therefore, activate different genes, make different proteins, and make these cells come out very different. And they don't only make the difference between cell types, they can actually even regulate entire programs and make entire organs. An extreme example of this that when I first heard it really blew my mind, so I want you to take this with well, I think it's a very striking example of this, is that people were looking in fruit flies at transcription factors. And they found a transcription factor, so one of these proteins, that is necessary for making an eye. This transcription factor, this protein, is called PAX6. And so it binds the DNA and it activates some genes. And because of that, the fly is able to form its eye. If it doesn't have this transcription factor, the eye is not there. It's not made. So eyes are very peculiar structures. We have eyes, right? And if we look at the eye of a fruit fly, it actually looks nothing like our eye. If we look throughout biology, the diversity in eyes is actually as extreme. Our eyes, the eyes of a fruit fly, the eyes of a planarian, or of a squid, they look nothing like each other. And they don't even function in the same way. So our eyes have a lens and we have a retina, and so we make an image on the back of our retina, and that allows us to see and to see pictures. You want to?
I heard that when your eyes take an image, it actually takes it in upside down.
That is exactly right. Yeah. Yeah, and your brain is able to decode that. Your brain doesn't know any better, so the image is always upside down, and so it knows how to read that. That is true. So we make an image. Flies actually also make an image, but they have lots of little camera, little eyes. Animals like planarians actually don't even make an image. All they see is whether there's light or not. They have no way of reading where that comes from. So these are very different shapes of eyes and very different functions of eyes. But remarkably, if we look at this gene at PEC6 that is important for making an eye in a fly, and we look at the sequence that makes this transcription factor, it turns out that that is actually conserved. So if we now look in a totally different model, in a mouse that doesn't have this PEC6 gene, it also cannot make an eye. And actually, it's even a known condition in some humans where they don't make a correct eye because they have a mutation in PEC6. So this same transcription factor, this same protein, is responsible for the making of an eye in a wide variety of organisms. It's even a little more crazy. We can take the PEC6 from a mouse, and make it go on somewhere else in a fly, for example, in its leg, and this fly will now make an eye in its leg. So it's really a master regulator. So having this one protein puts on a whole program and is hugely important for forming tissues and forming structures. And this is very widely conserved. And what this actually teaches us is that this variety in biology, all these things that look so different, that look like entirely new things, are actually probably not new. That all these things that look very different are not so much new inventions, but are rather variations. It's more tinkering with programs that are there, changing them a little bit. And this allows us to go from one type of eye to a different type of eye, but still the same basic principles are underlying this. And so that the same regulatory principles and the same regulatory mechanisms and molecules are used again and again and again in all these different organisms, even though they look completely not like each other. And that means that we can now actually learn from these organisms. If we want to learn how to do something that we cannot do, we can look at another organism and ask that is able to do this and ask, how are they doing this? And well, that is actually what we're doing in my lab. We're looking now at organisms that are outside of this standard set and asking, how can we learn from them? For example, can we learn how to live longer and healthier, which is something that I think nobody will object to. It seems like a nice idea. Because we all know that we, over time, do what we call age. Some of our tissues are not functioning as well anymore, and we have a limited lifespan. We get about 100 years, and that's roughly the time we have. But there's organisms out there that handle that completely differently. And that's the one that I want to introduce to you now. So this is a flatworm, Schmittea mediterranea, we call him. He comes from around the Mediterranean Sea. And so they live in freshwater streams. And these are actually organisms, they live in many freshwater streams. If you look here in little streams, in brooks, you might find similar flatworms around here. And you can see they can swim around. They freely swim around. Some of you may have seen them on the microscope outside in the demos. They clearly have some direction, so they have eyes, and you can see that they swim always after their head first. They also, we can see, have a pharynx in the middle of their body. So they don't have their mouth in their head, they have the mouth in the middle of in their belly button, basically. And their mouth is also their butt, so food goes in and out. They also have a lot of other structures. So they have, for example, a pretty big brain. And they have an intestine or gut that allows them to digest all kinds of things. this is the structure that I told you about, the pharynx, the tube that they use to eat through. And they also have muscles all around. They have structures that help them to secrete water, so to keep homeostasis, that are a little bit like our kidneys. And so they have all these different features. So even though they look pretty simple, they actually have a lot of the structures that we have too. But the most remarkable thing is their ability to heal. And you probably realize we have an ability to heal, right? If you have cut your finger, if you've ever done that, you know that this will repair, right? It will heal again. Or if you've ever seen somebody who's broken their arm, this will actually grow back together. And there's some other organisms that are actually pretty good at this, that can make fins or fishes or things like that. But for most animals, if you would chop off their head, that would kind of be game over. And I encourage you to not try this. For most animals, this is not a good idea. The one exception, I'd say, is Planarians. We can chop them into pieces, and both pieces actually just move on with their life. And this is not something remarkably cruel we do to them. I just want to give that as a warning. This is actually something they do themselves. So when they want to multiply, one of the ways they have to do that is to adhere their tail to the surface and crawl forward and tear themselves into two pieces. So this is something they naturally do. We're just helping them a little bit. And so this is what that looks like. So here's an intact Planarian, and here we've cut him into two pieces. And over the course of about 10 days, each of these pieces regenerates a full worm. So it makes the worm whole again. First, it just kind of closes up the wound, and then it starts to make this white tissue that we call blastema. So these are all newly made cells that the worm makes, and depending on what is lost, it will make the tissues that are necessary to make a whole worm again. So if it has lost its head, like this one, it will actually make a new head. You can see that it's making new eyes, it's making a new brain, tying that in with its body, and it will be fine ever after. And the one that still has a head now has to make a new pharynx and make a new tail, and they're able to do that. So you might wonder if this is the only way they're able to do that, or whether this goes a little further than that. And you're not the first one to wonder this. Actually, already around 1900, Thomas Morgan was wondering how small can you cut a worm, and can it still regenerate? So how small can you go? Can you cut it in different orientations? Can you cut it like that? Can you cut it in little segments? Can you even cut a little piece from its side? And the answer to all of those is yes. So it doesn't matter what you cut, how you cut them, where you cut them. As long as there's a little bit of tissue left, they can make a worm out of it again. And they don't do it just once. You can do it again and again and again and again, and they will keep making a new worm out of it. Do you have a question?
This animal is called the just like that. Basically, when it feels threatened, it redoes its life cycle and turns into that thing that sticks to the ground, and then things go down.
Absolutely. There's more organisms, so they're not unique in that sense. There's more animals around that can do this. They just happen to be not within our set of model systems that we picked, so the little puzzle pieces that we picked to start out with. They weren't like that, but actually when we look around, there's many more that can do this. Yeah, absolutely. So it's not unique. And that also means, again, that it's not something that other organisms cannot learn, or that cannot, adjust or try to incorporate. It is something that is universally there, except that many animals can't do it anymore, have lost the ability to do it. And so the question, of course, is, well, it's great that they are able to do this, but how? And what can we learn from this? Well, so the first part of the answer to that is that I told you about how cells are able to divide, how they go through a cell cycle. And we have a lot of cells that are able to do that in our bodies. Planarians have even more of them. They have a lot of cells that are here stained in pink that are able to divide, and that also constantly divide and constantly make new cells. If we would take those away, so there's things we can do to them. We can irradiate these animals so that they don't have dividing cells anymore. So here we can see how many dividing cells there are in the beginning. If we irradiate them, they go away really quickly. And then the worms also can't regenerate anymore. So these stem cells, as we call them, these dividing cells are essential for their ability to regenerate, for them to be able to heal and to keep living. What is quite unique about these stem cells is the phase that they're in. So I mentioned to you that when we make an organism, we start out with this one cell that we call the zygote, where the egg and the sperm have come together, and that divides into many, many cells and eventually makes all these different tissues. So that one cell is able to make anything, and we call that pluripotent. It means it has lots of possibilities. And that is a state that often is only there in embryos. So these cells can still make anything they want. But over the course of their development, these cells become more and more specialized. So they start to shut down some of the genes that they're not going to need. And eventually, when they are fully differentiated, when they have made a certain tissue, they actually can't go back anymore. They can't go and make something else. The Planarian stem cells now actually are still pluripotent. So whereas we don't have pluripotent cells when we are, born after birth, Planarians do, and they keep these cells around all the time. So they have a large population, a large group of cells that can still make anything. And they also maintain this very actively and dynamically. So if they need to make more tissue, so if they need to heal, they'll make more stem cells temporarily, they'll make more of them, and then make all the different tissues out of them. And then once they're done with that, they'll go back to their normal state. On the other hand, if we give them a little bit of radiation and they have fewer stem cells, also they will go back to the number that they used to have before. So they'll constantly be remaking and balancing their stem cells. Kind of a unique thing is that they also don't grow out of control. So they never make too many of them, which is something that we're very jealous of, that they are able to control these cells very carefully. So then after you have so many stem cells, how do they ever know what to make? Because, that's not really, the stem cells in the head have to make something different from the stem cells in the tail. And what turns out to be the case is that actually in Planarians, they constantly signal to their stem cells where they are. So they're kind of radio towers on different positions in the animal that constantly signal you're close to the head, or you're in the tail. And that is what is shown here. So these are ways to visualize how these genes, these signals are expressed, where these signals are highly concentrated. And so you can see that there's this signal, for example, here, is all in the tip of the head. So this is signaling to the stem cells that are receiving this signal saying you're in the head, you might want to make a brain. These ones here are all the way in the tail. And so they're signaling to the stem cells, don't make brain, this is the wrong place to do it. Some of them are close to the pharynx or are around the edges. And so the stem cells constantly listen to these signals and thereby respond to where they are and what they probably should be making. And so when we amputate a worm and cut this off, one of the very first things these animals do actually is make these signaling centers again, make these radio towers. Let me see, there it is. So this is the signaling center, for example, in the head. And we can see here a wound. We have one piece that has to make a tail and one piece that has to make a new head. And when we look at the expression, like the concentration of this signaling factor at the wound, we see that this one actually doesn't increase it doesn't really change because it has to make a tail. This one of the very first things it does is make this signaling center, make this radio tower again that's saying, here should be a head. And stem cells that are around here, you might want to think about making a brain. And then once they've done that, they actually fill in the blanks in between and fill in whatever else they need to make. Another thing that we've learned from Planarians is that not only do their stem cells listen all the time and listen to what they should be making, they're also really neat. They clean up their own mess. So once the cells have divided and have made different tissues, after a while the tissues get older and the tissues don't work so well anymore. And there is a system in cells that we call apoptosis, which is a way of killing the cells off and saying we're going to get rid of them now. Planarians are very good at that. They use that not only in cells that are getting older, but also when they are regenerating. So you could imagine, for example, that if we have this piece here that has to regenerate, it has now a gigantic brain, and it actually has very little gut, and it doesn't have a pharynx. So what it's doing over the process is actually killing off a lot of cells, saying, I don't need these anymore. There are too many. I can use this energy for better things. And they kill these cells off and then use the energy to make the cells that they actually need. So they're constantly monitoring how they're doing and whether their cells are in good shape and whether they're still necessary. And the last thing that we were learning, that we're very interested in my lab, is I told you about how there is so much in the genome, how there's so many bases in our genome, so many letters, and only some of them are genes. They're quite limited, actually. There's a lot of information that is in between that we don't really understand very well. There's also a lot of genes in every cell that should not be active. And we can consider that transcriptional noise. They should not be expressed, so a lot of these pieces of DNA, there are no genes, they shouldn't be making RNA, and they should just be quiet and be there. And that is another thing that planarians turn out to be very, very good at, is that they are constantly suppressing noise. And this is a system that we're visualizing where actually all of the planarian cells have active noise suppression that is here shown in this green staining. And if the cells don't have that anymore, they're starting to make all this kind of noise, and these cells don't function very well anymore. And this is something that is quite unique, that they're constantly suppressing this. And here we have actively tried to disrupt it to see what goes wrong. And we find that indeed they're not functioning as well anymore at that point. So we're using planarians as a model, as a way of learning about how to make new tissues and how to keep these tissues healthy forever, because this is what planarians are extremely good at. And we're studying all these different aspects. We're asking about these pluripotent cells, these cells that can make anything. So what is unique about them? How is it that planarians can keep those and that we can't? And how are they regulated? What are the molecules that are important for keeping these cells in good shape and for keeping them stable? We're also asking about the cells that are coming from them, like how flexible are they? And how are they making all these different tissues? What does that mean to go into all these different directions? How do they know what to do, and how do they integrate all these different signals? And so with that, I want to thank the brave people in my lab who helped me to figure all these things out. You've met several of them at the demo, and they're also going to be around to answer more questions. Francine Nyoseko, Michael Zelko is here, Danyan Lee, and Yan Chen is also here. So they'll, and here you can see a little image of us in the lab working on all these different questions. And so with that, I would be delighted to take any questions that you have, and give the last image to the worms, the stars of this show.